session number 2, we're going to move to the topic of clinical trials for EHE. And we're going to start off with Dr. Wagner from the Dana-Farber Cancer Institute, who's going to be talking to us about SARC 046.
All right, thank you very much. Give me a second just to share my screen. All right, can you see my slides?
Slides are there and look great, and your audio is great.
All right, excellent. Well, thank you for the introduction and for giving me the opportunity to highlight this study that actually just opened just about a month ago. And I'm going to talk about a clinical trial using Nab-Sirolimus, which is a drug for treatment of EHE. These are my disclosures. There we go. So as you just heard, so I'm not going to go into the background of the fusion and sort of how we think it drives EHE, but as you all know and just heard, the EHE tumors are characterized by very characteristic fusions of sort of alterations in the genes that then allow for the tumor cells to grow in an abnormal way. The most common one is a fusion of two genes, WWTR1 and CAMTA1, which is by far the most common. And then a more rare subset has a different fusion, but involving genes in a similar pathway, and that fusion is with YAP1 TFE3. And both of those genes impact a signaling pathway called the Hippo signaling pathway. And what that does is it then leads to activation of a number of different pathways within the EHE cell that would otherwise not typically be activated or are activated in just a less controlled way. Nab-Sirolimus, the drug that we're using in this clinical trial, is specifically targeting a pathway called the mTOR signaling pathway. And work by a number of different folks showed that the presence of the classic EHE fusions impact signaling through this mTOR pathway, and that's been shown in models in the laboratory. And in a model trying to actually mimic EHE in mice, treatment with similar versions of Nab-Sirolimus, but different drugs essentially targeting the same pathway, actually slowed down tumor growth. And when those tumors were removed, it confirmed that the drug was doing what the folks who did those experiments thought that it was doing by blocking signaling through that protein mTOR. It's been used, so sirolimus is a drug that's actually been around for a very, very long time. Its regular use is as an immune suppressant in patients who have solid organ transplants. But because mTOR signaling is relevant in actually a lot of different cancers, there have been a number of studies in other cancers. And then because of the data that has evolved in EHE, there are a lot of clinicians who will treat patients with EHE with drugs that target the mTOR signaling pathway. Sirolimus is, because it's been around so much and is relatively cheap, the one that's most often used. And the group in Italy, specifically out of Milan, published their experience treating what for EHE is a large number of patients with the drug sirolimus. So what they've published was all looking backwards. So that's what retrospective studies mean. And the largest group or cohort of patients that they published was a group of 38 patients, importantly, who all had disease that was actually growing when they started the sirolimus. The average amount of time that patients were on the drug was about 13 months, and about 10% of patients had their tumors shrink by at least 30% or more. And these pictures are just one example of a patient who had a fairly large tumor in the liver. So these are 4 different CAT scans at different time points. The time point is just below the picture. And what you're looking at here is, so if you can see the cursor, what I'm hovering over is the liver. And this darker spot within the liver is the EHE tumor. And you can see that it kind of starts fading away at 3 months, and then even more so at 12 months, and then even more so at 18 months. So it's a slow response. So that's almost or basically a year and a half, but uh, clearly benefit with the sirolimus treatment. So what is Nab-Sirolimus? Nab-Sirolimus is just a different version of sirolimus. The key alteration is that there's a protein called albumin that is actually bound to the active drug, the sirolimus. And what that does is it changes the distribution of the drug in the body. So what we think is that it achieves a higher concentration of the active drug inside the tumors, preferentially, which in theory would also increase the anti-tumor, anti-EHE activity compared to what we see with just the oral version or the pill form of sirolimus, which then would have to be absorbed through the gastrointestinal tract. One other potential benefit with the IV albumin-bound version, the Nab-Sirolimus, is that we don't have to monitor drug levels, where sirolimus is a drug that can actually be pretty tricky to give, even though it's a pill. We have to monitor patients' levels in the blood, so it just requires a lot of checking and titration of the dosing. Nab-Sirolimus has already been studied in people, and it's actually FDA approved for another ultra-rare sarcoma called PEComa. One important thing to at least note is that even though the study that led to the approval for PEComa didn't allow for patients to have prior just regular oral sirolimus, there was an expanded access program that uh, was open for a while after the initial study was completed and before the FDA approval was achieved. And that didn't have that same restriction. And there were actually some patients who had sirolimus in the past who had then had benefit still with Nab-Sirolimus. So there's at least some suggestion that Nab-Sirolimus is arguably better than sirolimus, and that's something that we'll hope to figure out in this clinical trial. So what is this study specifically? Everyone who goes on the study will get Nab-Sirolimus, and the way it's designed is a two-stage design. So that means that we will start enrolling just 12 patients, and if there's no activity, then we would basically just stop the study because we don't want to enroll more patients for a drug that's not doing what we think it's going to do. Where if out of those first 12 patients, one person has at least a 30% reduction in the tumor size, then we would open up the second stage of an additional 25 patients for a total of 37, what we say evaluable patients. There is an allowance just in case there are patients who, for whatever reason, either have to come off before scans or aren't meeting the protocol eligibility, which I'll show in a few slides, where we're enrolling up to 41 total patients on this study. The treatment is a relatively intensive treatment. So like I said, it's an IV drug, and it's given 2 out of 3 weeks, and that's given continuously. So for someone who goes on this study, if they were to start, I'll say today, then they would come back next week, get another infusion, and then have the next week off, and then the next week start that cycle all over again. So it does require a large number of visits into the clinic just to get the treatment. We have the following sites, so basically 5 sites, which is turning into 6 just for logistical reasons. But these are the sites that it is planned to open at. It's already open at Dana-Farber, where I am, and it just opened at Memorial Sloan Kettering in New York. Actually, this afternoon, I have a virtual visit with Stanford. So they're very close to opening. We also already scheduled the site initiation visit for WashU, and then Colorado, I think, is close behind as well. MGH is also planned to open, and we're just going through some logistical issues for that. One other thing to note, and this sort of piggybacks on Dr. Rubin's talk and his emphasis on having an expert pathologist review the pathology, where even though all of these centers have expert pathologists, that doesn't necessarily mean that the initial pathology was reviewed at any of our study sites. So we are requesting archival tissue, meaning tissue that's already been collected, and it's going to be reviewed by Dr. Papke at Brigham and Women's Hospital just to confirm the diagnosis. Um, this is an incomplete list of the inclusion criteria, but lists sort of the key points where, of course, everyone has to have confirmed EHE. We are not requiring that central confirmation prior to enrolling or prior to starting treatment, but we at least want to see a pathology report that very clearly says epithelioid hemangioendothelioma. Patients have to have measurable disease, which essentially just means disease that you can see and measure on a CAT scan. And there are very specific guidelines that are pretty widely used for clinical trials that we're also adopting. This study is only for adults who have to be at least 18 years old. Performance status is just the way that we measure someone's general overall health. So basically, if you can be up and moving around and doing most of your daily activities as normal, then that is sufficient to enroll. And then there are certain lab parameters that also have to be met, again, because we don't want someone who's so sick that they might need either another treatment or that it might sort of compromise the study if there's really no expectation of benefit. There are some things that can't be present. And I guess to the same point of you can't be too sick. So if someone has an active infection that's uncontrolled, then that would have to sort of be resolved before going on to a trial. You can't have had a big surgery within 4 weeks from going on trial. A biopsy is okay, but it can't be surgical. Um, there has to be at least controlled disease if there's anything in the brain or central nervous system, and you have to be at least a year out from having a heart attack, although hopefully people have just generally healthy hearts going in. And then there's a specific list in the protocol, which if you talk with your oncologist, they can make sure that you would otherwise be eligible. The main goal of our study is, of course, we want to see clinical activity, and that can be actually defined in a number of different ways, where one way to view it is actually shrinking the tumor, which is how this study is designed. But if you think about a growing tumor that then becomes stable, even if it doesn't necessarily shrink, that's also a very clear sign of benefit. The reason we're not using what we call progression-free survival or just delayed growth as our primary endpoint is because so many people with EHE will require treatment, but not necessarily have rapidly growing disease. And because this tumor can be so indolent for so long, we are using the actual tumor shrinkage as at least the key thing that our statistical design was planned around. That being said, we are still going to look and see, are we prolonging the amount of time that people will be able to stay on one treatment before they have to change and things like that. And that's exactly what a secondary objective is. So even though the primary endpoint or primary objective of the study is to look at tumor shrinkage, we're going to see are tumors that were growing, not growing anymore? Are people still alive a year after stopping therapy? And then importantly, we're also asking patients to fill out a few different surveys. So one is called the EQ5D3L, which is a survey that was developed actually by one of the cooperative groups, so sort of a multi-institutional effort started in Europe to ask the question, what is your quality of life? And our hope is that patients' quality of life by going on the study will actually improve. And then because pain is such a significant symptom in patients with EHE, we added something called the brief pain inventory, which is an additional short questionnaire asking specifically about some more details about pain. And our hope is that we'll see patients' pain improves as well with treatment. So as I mentioned, the study schedule is pretty intensive. The treatment itself is given 2 weeks in a row. So for patients who are getting this treatment, they come in, get the infusion, go home, and then come back a week later to get another infusion. And then the third week is off. And then that cycle just keeps repeating as long as you're tolerating the drug and as long as the tumor is controlled. We are requiring that, at least for the first few infusions, patients actually see their doctor or their nurse practitioner or PA in the clinic, however the site is set up. But as the study goes on and as someone sort of demonstrates that they're tolerating the drug a bit better, then we don't require the clinic visits every time. The scan frequency is every 6 weeks for the first 36 weeks, and that's because we want to keep a fairly close eye on what is happening with the tumors. And then if they're growing, then obviously patients would come off. But then as patients get further into the study, eventually the frequency of the scans decreases to every 12 weeks. And then there's at least one visit after coming off of treatment just to make sure that there's no additional side effects or hospitalizations or anything else that might have come up that could be attributed to the drug. We have a few planned translational or correlative science objectives. So one of the main ones is what I already mentioned, is that we want to even just confirm that everyone who goes on actually has EHE. So that is going to be done in collaboration with the pathology team at Brigham and Women's Hospital. We're going to do sequencing to confirm the presence of, or see if we can at least detect any of the gene fusions that are characteristic for EHE. And then we're also going to look at some of the proteins that are important in the mTOR signaling pathway and see, are there any markers that might help us predict who would benefit from this treatment versus others. We're also doing a blood test, which has been shown out of the European, really the Italian experience, that a blood test called MIC1 or GDF15 level correlates with the level of disease activity, and they actually looked at that specifically in patients who are on the oral version of sirolimus. To get more information, I would say the best way is to actually talk with your oncologist or to contact any of the sites that are closest to you. So from that list that I showed before, and I think this is going to be public, or you can email this email here, which is the general email for the clinical trial team. It's sarc046@sarctrials.org. And thank you for listening, and thank you to the few folks, if you're on, who have already joined to this study, and anyone who will join in the near future. We very much appreciate the patient engagement and really can't do what we do without sort of your support, and we do it with the hope of improving just the lives and helping folks with EHE. So thanks so much.
Thank you very much. And we'll have you back for the Q&A in a bit. There are a few specific questions about the trial in the Q&A section. If you have a chance to get to it, that's great. If not, we'll try to come back to as many of those as possible in the Q&A session.
Okay, sounds good. Thanks.
Our next speaker is going to be Dr. Italiano from Institute. I'm going to mispronounce it, so I'll have him introduce the name of his institute here, talking about a TEAD inhibitor from a clinical trial for ODM 212.
So thanks a lot for the introduction. Actually, I'm working at Gustave Roussy in Paris. So I'm very happy to share with you some insights regarding a new therapeutic agent that is currently under evaluation in a study called the TIDAS, which is sponsored by by Orion. what you have to know first, before giving you the detail, is that this study is what we call a phase 1 study. So a drug pretty new in the clinical setting. So it's what we called early stage research. And so far, only a very few number of patients with EHE have been included in the study. So I will present you some preliminary data, but the study is still ongoing, and so the results of this study may change in the future. Again, this drug is an experimental drug, so it's not an approved treatment. And if you wish to participate to this study, the best approach is to discuss with your oncologist. So you know the rarity of this disease. Behind the clinical aspect, there is a complex biology that can be actually targeted by this new drug called OTM-212, which was designed specifically to inhibit the YAPTAZ pathway, which play an important role in tumor progression of EHE. So, what is the TDES study? Again, this is an early stage study, a phase 1 study, including adult patients with advanced tumor, characterized, and in this study, we include patients with tumors characterized by a strong activation of the EPO pathway, and this includes epithelioid hemangioendothelioma. So in this study, the drug which is evaluated in terms of efficacy and safety is OTM-212, which is an oral drug taken every day. And so in this study, the objectives are to assess the safety of the drug, so evaluating the side effects in the patient, and also early signals of efficacy. So one key objective of the study is to define what is the right dose to administer to the patients. So meaning that not all the patients in the study have the same received the same dose of the drugs. We started very small dose, and then based on the safety profile, we have increased the dose of the drug in the upcoming patients. So, what is the mechanism of action of the drug? In epithelioid hemangioendothelioma cells, there is an activation of several pathways that promote tumor progression. And one of these pathways is the YAPTAZ pathway. And so basically, this drug, OTM-212, is designed specifically to block this pathway, which is involved in tumor progression. And so in this way, we can inhibit the proliferation and the survival of tumor cells. So what were the results of this study reported so far for what regard epithelioid hemangioendothelioma patients? 3 patients were assessed. All of them had clinical benefit. For one of these patients, the treatment is still ongoing more than 1 year after treatment onset. Actually, this was one of my patients, who is doing actually very well. And for 2 of them, we have observed a stabilization of disease for more than 10 months. In all these 3 patients, the side effects were quite manageable, and actually the drug was very well tolerated. So first example, so this was the first patient in the world receiving this drug for epithelioid hemangioendothelioma. This is one of my patients, a 59-year-old man with epithelioid hemangioendothelioma with metastasis in the liver, the lungs, and the bones. He was referred to me with progressive disease, and symptomatic disease actually. And so he had first first-line chemotherapy with doxorubicin, which actually had some effect in terms of clinical stability, but then he progressed, and so I proposed him to be included in the TDES study. So he started the drug, which was very well tolerated, and the first CT scan, as you can see here, show a very significant decrease of the liver metastasis. You can see here, so this is the liver. These are in here. You can see the metastasis at the beginning of the treatment, and you can see that a new CT scan performed a few months later show a very strong shrinkage of this lesion. So the patient is still on treatment and is doing very well. There are two other examples of clinical benefit of this drug. The second patient included in the study had an epithelioid hemangioendothelioma with liver and lungs metastasis. She received in the first-line setting pazopanib for treatments, but with no clinical benefit, and so there was a progression of the disease, and this is the reason why she was proposed to be enrolled in the study. She started the treatment, and again, the drug was very well tolerated, and there was a stabilization of the disease with a shrinkage of the lung lesion. For the third patient, a 61-year-old woman with epithelioid hemangioendothelioma in the liver and bones, again, she received pazopanib before to be enrolled in the study. And after progression on pazopanib, she was proposed to be included in the study. She received the drug at the same dose of the previous one. And again, this patient had very good tolerance with very limited side effects and clinical benefit with shrinkage in liver lesion. The clinical benefit is still ongoing. So the trial, this phase 1 study is available in several countries in the world, in Finland, in France, in Spain, in Switzerland, in the UK, and in the USA. You can find some information regarding the sites who are activated for this study on clinicaltrials.gov. And again, the best approach is to talk about your doctor of the possibility to be enrolled in this study. So what are the key take-home message? Clearly, and this is my personal opinion, this new drug represents real hope for developing a new efficient treatment for epithelioid hemangioendothelioma. But again, the study is ongoing and we have to wait its completion to be sure about the tolerability and the efficacy in this disease. And I have to thank all the patients and their family who trust us by consenting to participate in this important study for epithelioid hemangioendothelioma. Thank you very much for your attention.
Thank you very much for the exciting early details, understanding that early. We're now at our Q&A session. If we can have Dr. Wagner back, and I believe I'm going to be joined by Denise as well. And just one note to, based on a number of the questions we've been getting, we unfortunately are not able to give specific medical advice during this conference to patients with specific scenarios. They're very good questions you're asking. What I would recommend is to ask those questions to your clinician or your treating panel, including even copying some of the information that you're learning here today so that they have some context. But unfortunately, we can't go into that level of detail at the conference here. All right, so I think this is a question for both of you. We got questions from the viewers wondering that if someone's on the trial, and let's say they participated in the phase 1 trial for the TEAD inhibitor, they're participating in the phase 2 trial for the Nab-Sirolimus, do they get to stay on the drug through the trial and potentially afterwards is the question, especially if things are working well?
Yeah, so I guess answer first. So I guess the short answer is yes. So certainly if it's working well, then there's not a firm endpoint as long as we're able to continue. So patients would just continue on. And then honestly, even if someone has tumor growth or has to come off study, there is an allowance to potentially continue if it's felt that there's benefit, and that would of course require a discussion with your oncologist and with me and the study team just to sort of make sure that's a acceptable thing to do. But that is actually sort of written into the protocol that we could potentially continue even after progression.
And how about for the patients in the T trial?
Yeah, and it's the same for the TDS trial. Yeah, if there is clinical benefit, the patient can remain on treatment. Yeah.
Yeah. And then I'll just make a note, at least in the United States, and I believe this is true in a number of other countries, if a drug is already FDA approved for some other use, clinicians are allowed to use that drug off-label, even if it's not FDA approved for that indication. That's a decision between patients and doctors, but at least in the United States, there is some ability to use drugs sort of, even if there is not an FDA approval, if they are approved for something else.
And paid for, I'll add.
Yeah.
It's not a cheap
Yeah,
drug.
that's always the problem, is might not be paid for.
Yeah.
So both of these drugs that you have talked to us about today, there's sort of other forms or kinds of drugs either in the same class or similar in some other way. For patients that have either been on, say, oral sirolimus and are now considering Nab-Sirolimus, is there an advantage to one formulation versus another? Is that something that patients could consider? And then conversely, if a patient has perhaps been treated with a TEAD inhibitor of some kind before, is there an advantage or disadvantage or considerations about looking to enroll in a trial with a different drug that's a similar medication?
Yeah, so I think that's a good question and not always so easy to answer. But just using the mTOR inhibitors as an example, so there are a number of different mTOR inhibitors that are used in the clinic for various reasons, where Sirolimus is just the one that I mentioned. And I saw also there were questions about, are we allowing patients who have had prior mTOR inhibitors? So the short answer to that is yes. Part of the reason is because, at least in theory, we think that the Nab-Sirolimus can be better than some of the other versions. And that's really the main reason for doing the study. We don't know. I can't say that that's necessarily true, but that's what we're hoping to learn from the clinical trial. And then to the question of, can patients who have had any of these prior mTOR inhibitors, whether it's sirolimus or everolimus or something like that, enroll on the study? Yes. However, I think it would require a conversation with your oncologist about, is this the right trial for you? If you were already on one of those drugs, what was the reason for coming off? If your tumor just grew through sirolimus or a similar drug, then it really might not be the best clinical trial option for you. And similarly, if you had a lot of toxicity with drugs targeting that same pathway, I would expect a lot of that toxicity to come up with the Nab-Sirolimus as well. So that would be another thing to consider. So we do allow it, and I think when we do the final analysis, we'll look at patients who have had prior mTOR inhibition and those who haven't, and see if there's a clear difference. But I think it should be a conversation if this is even the right trial for you if you've had prior sirolimus versus other options out there.
Thank you. Dr. Italiano, did you have any thoughts about that with additional TEAD inhibitors?
Yeah, I agree. It's not there is no an universal answer to this question, but usually if there is a disease progression on a drug, on a specific drug, it's better to consider for the new line of treatment a drug with a different mechanism of action, because we can assume that some mechanism of resistance to a drug targeting a specific pathway could be associated with resistance to other drugs targeting the same pathway. So I think it's always better to, if this is possible, to consider an alternative mechanism of action in case of disease progression to a specific drug.
We've gotten a number of questions about access to the trials, especially crossing borders. You both presented the geographic areas where trials are being conducted in terms of cities and countries. If someone's from a different country, are they able to access these trials?
Well, in France, we cannot, we have a very conservative regulation, and so we cannot enroll in clinical study patients who are not affiliated to the French Social Security. So affiliation to the French Social Security is relatively easy for patients coming from countries from the European Union. But this could be very complicated for patients coming from countries outside the European Union.
And how about for the Sirolimus trial?
Yeah, so it's only planned to be open at the specific sites sort of across the country in the US right now. If someone has insurance coverage and can actually travel to those sites, then it would be possible, and we actually sort of built into the funding. So this is a study that's actually funded by the US Department of Defense. So they had a grant for rare cancers that we applied for and were successful in getting the funding for it. And in that grant budget, we included reimbursement for travel, which could include the actual traveling and hotel and various aspects of the hassle and travel that's required to get to one of these few sites. But it is a pretty intensive study schedule, so it would require a lot of traveling. We can't give the study drug somewhere else, even within the US. It's only available at the specific sites. And then also most of the study visits and the doctor's visits, the infusion visits and things like that are generally billed to insurance. So there could be cost associated with that. And especially for someone who doesn't have insurance coverage in the US, that would get very, very expensive, which I would imagine would be prohibitive for most people.
Thank you for sharing that. We are unfortunately at time for questions for this session. We really appreciate both of you joining and sharing your expertise and answering questions about the nuances of the trials you're running. They're very exciting to have these options available for patients.
Thank you.
Thank you.